Today I'd like to talk to you about sequencing the DNA of children with genetic conditions and then using the data for other purposes. But first, let's talk about what DNA is. DNA contains the instructions that tell our bodies how to grow and develop. Each person has a unique DNA sequence that affects many things about them, such as their hair and eye color, as well as their chance of developing some diseases. DNA is made up of a sequence of letters, A's, T's, C's, and G's. So what is genomic sequencing? Genomic sequencing is a technique that is used to read DNA. It finds the order of the letters of a person's DNA one by one. This DNA sequence can be referred to as genomic data. There are a lot of letters to be read, which means that genomic sequencing can produce a lot of data. Once the DNA sequence is obtained, the genomic data can be used by doctors and scientists to help identify the causes of genetic disease in patients. Doctors and scientists look for changes in the genomic data, called variants, that are different between people with diseases and people who are healthy. These changes might be very small, like spelling mistakes, or bigger, where large runs of letters are missing. Scientists often only look at some parts of the genomic data. Which parts they look at will depend on what symptoms the patient has. This means that for every patient, there is a lot of genomic data that won't have been analysed yet. You may be wondering why genomic data might be different from other health information. We inherit all of our DNA from our parents. We also pass our DNA on to our children. This means that we share some of the same sequence of DNA with our parents and children, as well as our brothers and sisters, aunts, uncles, and grandparents. So if a variant that causes disease is found in us, it may also be present in our blood relatives. This is different from health information that might be found through most other tests. Just because we have a variant in our DNA from birth that might cause a genetic disease, that does not mean that we will develop symptoms straight away. For some genetic diseases, we may not develop symptoms until we are a few years old. For others, we may not show signs of the genetic condition until we're in adolescence, early adulthood, or even much older, if at all. This means that looking at our genomic data can predict what kinds of conditions we might get in the future. Again, this is different from health information that might be found through other tests. We have told you we are interested in your views about storing genomic data, but why might we want to store genomic data from children with genetic conditions? The parents of some children who are thought to have genetic conditions are offered genomic sequencing. This is helping find the cause for their child's condition, which may lead to better treatments. It can also be helpful for parents to have this information when making decisions about having more children. Rather than throw away the data after the cause of the child's condition is found, the data could be stored for use in the future. This could be helpful if the child gets sick again with a different illness and the doctors want to look at the genomic data to see if they can find another cause. The data could be useful when the child becomes an adult as well. When they are ready to start a family, they might want to know if they and their partner carry any variants that mean they have a risk of having a child with a genetic condition. This is called carrier screening. They may also want to have screening to see if they are likely to develop another genetic condition later in life, like breast and ovarian cancer. So, what are some of the challenges of storing genomic data? One problem is that the digital files that contain a person's genomic data are very large. This can mean that it is expensive and difficult to store genomic data for large numbers of people in the one place. One way to deal with this could be for each research group, laboratory or hospital to store the genomic data from their own research study themselves, rather than having a more central storage facility that services many groups. Another solution could be for the data to be stored off-site in data servers and made accessible through cloud computing services. Cloud computing can put protections in place, like data encryption, that make data secure, although data breaches still occur. Another issue that can come up when data is stored at multiple different storage facilities relates to the format in which the genomic data will be stored. Laboratories can produce different types of genomic data files, which may not be compatible with each other. This could make it difficult for databases that want to access the genomic data to read them, which we'll discuss more in a moment. We've discussed data storage, but also told you we're interested in your views about data sharing. So why might we want to share genomic data from children with genetic conditions? Sometimes sequencing of a patient will identify a variant we don't know enough about, which stops us from being able to say for sure whether it is or is not the cause of the genetic condition. In these cases, it is helpful to share the information about the variant and also information about the symptoms on a database in the hope that other laboratory scientists or doctors may have a matching patient with the same or a similar variant. A match like this could confirm that the variant is actually the genetic cause. This might also help the other patient and future patients in the same way. While sharing genomic data to databases may be done for clinical purposes, like to find a diagnosis which could lead to a treatment or surveillance, this kind of data sharing could also allow researchers to study genomic data of patients in order to help identify new genetic causes of disease and to advance science. Data could also be shared with commercial companies, such as pharmaceutical companies, in order to assist them to develop new medications that are targeted to patients with particular genetic conditions. Government bodies, such as law enforcement agencies, may also want to access genomic data in order to compare it with their own databases to help them solve crimes. In the case of children with genetic conditions, accessing their data might identify family members or even the patient themselves when they are older. But what are some of the challenges of sharing genomic data? Some of the challenges relate to technical issues. As we mentioned earlier, laboratories can produce different types of genomic data files which may not be compatible with each other. This could make it difficult for the data to talk to each other within databases, almost like it was stored in different languages. If data is being shared, there are also challenges in making sure that only authorized people get access to the data. For example, if data is being shared for research purposes, you may want to make sure that only genuine researchers from particular research centers have access, and not someone without appropriate qualifications. Someone needs to make these decisions about who gets access, and who this might be will depend on how and where the data is stored. Other challenges relate to making sure that the privacy of the patient is protected. If data is being shared, it needs to be done in a way where information that may help to identify the patient, such as name, date of birth, etc., is removed. This may be very difficult for patients with rare disease, where there may only be a few patients with that genetic condition in the whole world. We also want to ensure that patients are not discriminated against, for example, in their ability to get insurance or a job because their privacy is not being protected. There are also legal challenges. Different countries or regions will each have their own legal requirements that must be met for data to be shared. This can cause issues if data is being shared across different countries or regions where this is the case. Another issue that relates to both data storage and data sharing is the need to obtain informed consent to do these things. So what is informed consent? Obtaining consent from someone just means that we're asking their permission to do something. When we say we want consent to be informed, it means that we want that person to understand what they are agreeing to. This will often involve providing them with information about the options available to them and also what the potential implications of their choices might be. In genomic sequencing, patients or their parents or legal guardians will often need to sign a consent form to show that they consent to the testing. We might also want to get consent from that child themselves when they become an adult to make sure they are still happy for the data to be stored and/or shared. It could be challenging to find them to ask their consent so many years later. There are a few different types of informed consent and ways of asking for it. Consent can be opt-in, where the person specifically needs to agree to something. In the context of genomic sequencing, this is often done by ticking a box on the consent form. Consent for some things may be implied, where specific consent for an action to be taken is assumed based on the context. For example, in a medical context, if a patient has agreed to receive medical treatment for an illness, it may be assumed that they also consent to their medical information being used to provide safe and effective treatment. Consent may be specific, where the consent provided by the patient relates only to a particular situation or project. Or it might be broad, where by giving consent they agree for their genomic data to be used for a much wider range of purposes. Consent is often static, which means it's obtained before testing is done and is not requested again. However, some research projects use dynamic consent, where participants have the option to change their consent preferences over time. Thanks for listening, and we look forward to seeing you in the focus group.